Chemical instability of RNA and the ubiquitous presence of ribonuclease make RNA extremely susceptible to degradation and more fragile than DNA. Today, let's see how to work with those precious RNA. Degraded RNA often leads to unsatisfactory results in downstream applications. Therefore, after RNA extraction, it is necessary to perform quality tests on RNA to determine whether it meets the requirements of various downstream applications, such as reverse transcription, qPCR, northern blot, cDNA library preparation, and cDNA labeling for microarray analysis. The yield of RNA is highly tissue-specific, determined by the abundance of RNA in different tissues and the difficulty of RNA extraction. Now, let's take a look at the following two methods to measure the concentration of RNA. Nucleic acid molecules contain conjugated double bonds in their purine and pyrimidine rings that can absorb ultraviolet light and have a specific absorption peak at 260 nanometers. Most proteins contain aromatic amino acids, which have the maximum absorption at 280 nanometers. In addition, some salt ions and organic compounds have an absorption peak at 230 nanometers. The nucleic acid concentration can be measured using a UV spectrophotometer, according to these physicochemical properties. In general, an OD260 of 1 is equivalent to 40 nanograms per microliter of single-stranded RNA. Hence, the concentration of the RNA sample is calculated as OD260 times dilution factor times 40 nanograms per microliter. In addition to the concentration of RNA, OD260-280 and OD260-230 can indicate the purity of RNA. Be careful, the solution pH can affect the measurement of odd value. For example, if DDH2O is used for elution, the solution is weakly acidic, which can increase OD280 and decrease OD260-280. Fluorescent dyes can specifically bind to target molecules and emit fluorescence under the excitation of a light source at a specific wavelength. Then, the fluorometer reads the received fluorescence value and converts it into a concentration value for accurate quantification of DNA and RNA. UV spectrophotometry and the fluorometric method have specific advantages and are recommended to be used together. UV spectrophotometry can indicate the purity of nucleic acids. The fluorometric method delivers higher specificity and sensitivity, which is suitable for downstream applications requiring precise quantification, such as next-generation sequencing, and for precious samples at low concentrations. The most common method used to assess the integrity of total RNA is to run RNA on an agarose gel. Before reading the electropherogram, you need to understand the term sedimentation coefficient, which is a measure of the rate at which biomacromolecule sediments in a centrifugal field. A higher value indicates a larger molecule. The electropherogram shows 3 RNA bands from top to bottom. The top 2 bands are bright, clear, and sharp. For example, the edges of the bands are clear. For general animal samples, 28S, 18S, and 5S RNA bands can be observed. If the extraction process involves column loading or TAP plus Li-Cl treatment, the 5S band may be darker or absent. For samples from insects or mollusks, there is only one distinct band. For general plant samples, 3 bands can be observed, 25S, 18S, and 5S. For general prokaryotes, 3 bands can be observed, 23S, 16S, and 5S. Theoretically, 28S:18S is 2.7 to 1. However, a 2 to 1 ratio has long been used as a criterion for identifying intact RNA. In fact, almost all RNA extracted from most samples falls short of the 2 to 1 ratio. Using the DNA marker as the control, if the 28S band at 2 kilobits and the 18S band at 0.9 kilobits are clear and 28S:18S greater than 1, the integrity of RNA can meet the requirements of most experiments. No redundant bands. There are 2 possibilities for miscellaneous bands: presence of DNA contamination or where multiple bands can be observed for the tissue type. For example, samples containing a large number of chloroplasts, such as plant leaves. The Agilent 2100 bioanalyzer assay provides a precise concurrent digital assessment of RNA integrity, purity, and degradation via a lab-on-a-chip microfluidics platform, and can be used as an alternative to conventional gel assays. Its electrophoretic trace and RNA integrity number are used to indicate RNA integrity. Agilent 2100 bioanalyzer trace. If the nucleic acid is intact, the baseline is flat. If the nucleic acid is severely degraded, the baseline is not flat, and more peaks indicating degradation appear. The value reflects the RNA integrity. Within the range of 0 to 10, a larger value indicates greater RNA quality and integrity. The larger the RIN value, the flatter the baseline and the sharper the main peak. In general, RIN greater than or equal to 5 indicates good RNA integrity, and RIN greater than or equal to 8 indicates perfect RNA integrity. The figure below shows a typical trace of a eukaryote generated by Agilent 2100 bioanalyzer. There are commonly 5 peaks in a typical electrophoretic trace of a eukaryote generated by Agilent 2100 bioanalyzer. However, not all eukaryotes have these peaks. For example, the organelles of plant leaves also contain ribosomes, and the 3 small peaks before 18S are for chloroplast RNA.